Chapter 13 Photosynthesis in Higher Plants. All animals, include human beings, depend on plants for their food. Have you ever wondered from where plants get their food? Green plants, in fact, have to make or rather synthesize the food they need, and all other organisms depend on them for their needs. The green plants make or rather synthesize the food they need through photosynthesis, and are therefore called autotroph. You have already learned that the autotrophic nutrition is found only in plants, and all other organisms that depend on the green plants for food are heterotroph. Green plants carry out photosynthesis, a physiochemical process by which they use light energy to drive the synthesis of organic compound. Ultimately, all living forms on earth depend on sunlight for energy. The use of energy from sunlight by plants doing photosynthesis is the basis of life on earth. Photosynthesis is important due to two reasons. It is the primary source of all food on earth. It is responsible for the release of oxygen into the atmosphere by green plants. Have you ever thought what would happen if there were no oxygen to breathe? This chapter focuses on the structure of the photosynthetic machinery and the various reactions that transforms light energy into chemical energy. 13.1 What do we know? Let us try to find out what we already know about photosynthesis. Some simple experiment you may have done in the earlier classes have shown that chlorophyll, green pigment of the leaf, light and CO2 are required for photosynthesis to occur. You may have carried out the experiment to look for starch formation in two leaves, a variegated leaf or a leaf that was partially covered with black pepper and exposed to light. On testing these leaves for the presence of starch, it was clear that photosynthesis occurred only in the green parts of the leaf in the presence of light. Another experiment you may have carried out where a part of leaf is enclosed in a test tube containing some KOH soaked cotton which absorbs CO2 while the other half is exposed to air. The setup is then placed in light for some time. On testing for the presence of starch later in the two parts of the leaf, you must have found that the exposed part of the leaf test is positive for starch while the portion that was in the tube tested negative. This so that CO2 was required for photosynthesis. Can you explain how this conclusion could be drawn? 13.2 Early experiment. It is interesting to learn about those simple experiments that led to a gradual development in our understanding of photosynthesis. Joseph Priestley 1733 to 1804 in 1770 performed a series of experiments that revealed the essential role of air in the growth of green plants. Priestley, you may recall, discovered oxygen in 1774. Priestley observed that a candle burning in a closed space, a bell jar, soon gets extinguished. Similarly, a mouse would soon suffocate in a closer space. He concluded that a burning candle or an animal that breathed the air both somehow damaged the air. But when he placed a mint plant in the same bell jar, he found that mouse stayed alive and the candle continued to burn. Priestley hypothesized as follows: plant restored to the air whatever breathing animals and burning candle remove. Can you imagine how Priestley would have conducted the experiment using a candle and a plant? Remember, he would need to rekindle the candle to test whether it burns after a few days. How many different ways can you think of to light the candle without disturbing the setup? Using a similar setup as the one used by Priestley, but by placing it once in the dark and once in the sunlight. Jan Ingenhouse 1713 to 1799, so that sunlight is essential to the plant process that somehow purifies the air fouled by burning candle. by burning candle or breathing animals. Engine house is an elegant experiment with an aquatic plant showed that in bright sunlight small bubbles were formed around the green parts while in the dark they did not. Later he identified these bubbles to be of oxygen. Hence he showed that it is the only green part of the plants that could release oxygen. It was not until about 1854 that Julius von Sack provided evidence for production of glucose when plant grows. Glucose is usually stored as starch. His later study showed that the green substance in plant is located in a special body within plant cell. He found that the green parts in plant is where glucose is made, and that the glucose is usually stored as starch. Now consider the interesting experiments done by T.W. Engelmann in 1843 to 1909. Using a prism, he split light into its spectral component and then illuminated a green alga cladophora placed in a suspension of aerobic bacteria. The bacteria were used to detect the sites of O2 evolution. He observed that the bacteria accumulated mainly in the region of blue and red light of the split spectrum. A first action spectrum of photosynthesis was thus described. It resembles roughly the absorption of spectra of chlorophyll A and B discussed in section 13.4. By the middle of the 19th century, the key feature of plant photosynthesis were known, namely that plants could use light energy to make carbohydrate from CO2 and water. The empirical equation representing the total process of photosynthesis for oxygen evolving organism was then understood as where CH2O represented a carbohydrate. Example glucose, a 6 carbon sugar. A milestone contribution to the understanding of photosynthesis was that made by a microbiologist, Corn, Cornelius von Neil, 1897 to 1985, who based on his study of purple and green bacteria demonstrated that photosynthesis is essentially a light dependent reaction in which hydrogen from a suitable oxidizable compound reduces carbon dioxide to carbohydrates. This can be expressed by in green plants. H2O is the hydrogen donor and is oxidized to O2. Some organisms do not release O2 during photosynthesis when H2S instead is the hydrogen donor. For purple and green sulfur bacteria, the oxidation product is sulfur or sulfate depending on the organism and not O2. Hence he inferred that the O2 evolved by the green plants comes from H2O, not from carbon dioxide. This was later proved by using radioisotopic technique. The correct equation that would represent the overall process of photosynthesis is therefore what C6H12O6 represent glucose. The O2 released is from water. This was proved using radioisotope technique. Note that this is not a single reaction but description of a multi-step process called photosynthesis. Can you explain why 12 molecules of water as substrate are used in the equation given above? 13.3. Where does photosynthesis take place? You would of course answer in the green leaf or in the chloroplast based on what you earlier read in chapter 8. You are definitely right. Photosynthesis that does takes place in the green leaves of plant, but it does so also in other green parts of the plant. Can you name some other parts where you think photosynthesis may occur? You would recollect from previous unit that the mesophyll cell in the leaves have a large number of chloroplasts. Usually the chloroplasts align themselves along the walls of the mesophyll cell such that such that they get the optimum quantity of the incident light. When do you think the chloroplast will be aligned with their flat surface parallel to the walls? When would they be perpendicular to the incident light? You have studied the structure of chloroplast in chapter 8. Within the chloroplast there is a membranous system consisting of grana, the stroma lamella, and the matrix stroma. There is a clear division of labor within the chloroplast. The membrane system is responsible for trapping the light energy and also for the synthesis of ATP and NADPH. In stroma, enzymatic reactions synthesize sugar, which in turn form starch. The former set of reactions, since they are directly light driven, are called light reaction. Photochemical reaction, the latter are not directly driven but are dependent on the product of light reaction, that is ATP and NADPH. Hence, to distinguish the latter, they are called by convention as dark reaction, carbon reaction. However, this should not be construed to mean that they occur in darkness or that they are not light dependent. 13.4 How many types of pigment are involved in photosynthesis? Looking at plants, have you ever wondered why and how there are so many shades of green in their leaves? Even in the same plants, we can look for an answer to this question by trying to separate the leaf pigment of any green plant through paper chromatography. A chromatographic separation of the leaf pigment shows that the color that we see in leaves is not due to a single pigment but due to four pigments: chlorophyll A, chlorophyll B, and carotenoid. Let us now see what roles various pigments play in photosynthesis. Are photosynthesis pigment photosynthesis Pigments are substances that have an ability to absorb light at a specific wavelength. Can you guess which is the most abundant plant pigment in the world? Let us study the graph showing the ability of a chlorophyll A pigment to absorb light of different wavelength. Of course, you are familiar with the wavelength of the visible spectrum of light as well as Vibgyor from figure 13.3a. Can you determine the of wavelength color of light at which chlorophyll A so the maximum absorption? Does it so another absorption peak at any other wavelength? True. If yes, which one? Now look at showing the wavelength at which maximum photosynthesis occurs in a plant. Can you see that the wavelength at which there is maximum absorption of chlorophyll A, that is in the blue and the red region, also shows higher rate of photosynthesis? Hence, we can conclude that chlorophyll A is the chief pigment associated with the photosynthesis. But by looking at figure 13.3c, you can say that there is a complete one-to-one overlap between the absorption of spectrum of chlorophyll A and the action spectrum of photosynthesis. These graphs together show that most of the photosynthesis takes place in the blue and red region of the spectrum. Some photosynthesis does takes place at the other wavelength of the visible spectrum. Let us see how this happens. So, chlorophyll is the major pigment responsible for trapping light. Other thylakoid pigment like chlorophyll B, xanthophyll, and carotenoid, which are called accessory pigments, also absorb light and transfer the energy to chlorophyll A. Indeed, not only enable a wide range of wavelength of incoming light to be utilized for photosynthesis, but also protect chlorophyll A from photooxidation.